Good afternoon. There's two right there in the middle.
Should we go there?
It's up to you. Taylor's saving seats apparently.
There's like a laptop.
Taylor just gave me a dirty look. I'm not saving seats. Leave me alone. Those two, right? It's just Abby, right? Abby? Getting it. I'm figuring it out. Good afternoon. Oh, I remembered it's afternoon. It might be morning to you. That's a problem. If you woke up 2 hours ago, you need to get yourself checked out. All right, so let's get started because we got a lot to do today. Got to go through erythrocytes and so on. PowerPoint's up, so you're good to go. All right, so we're going to talk about erythrocytes today, the structure and their function. We're going to talk about leukocytes, which you all covered pretty in-depth in lab, so I'm not going to go over them too much. All right, I will go over the basic structure and their importance, and then we'll go on from there. Okay? And then hopefully we'll get to the beginning of hemostasis. We'll go through blood typing as well, and hopefully we'll get through the beginning of hemostasis. If we don't, we don't. We'll finish that on Monday. All right, so erythrocytes, also known as red blood cells, okay? Which are, it's a misnomer because they're actually not cells anymore anyways. All right, this is a great, erythrocytes is a great illustration of the relationship between structure and function, okay? The overall function of erythrocyte is to do what? Transport oxygen, okay? And we're going to talk about the structure of the erythrocytes with that in mind, okay? That their job is to transport oxygen. So their structure is going to be modified such that they are the most efficient at doing that job, okay? Understand that? Okay, good. These are small, flexible cells, okay? You saw them on the blood smear. They look like the little donuts, okay? They are considered or they are described as biconcave discs. So on both sides they have an indentation. What do you think the benefit is of having that structure as opposed to just a big circle? All right, so they hold oxygen. Remember what they're doing. Remember, keep in mind their function is to do what? Transport and deliver oxygen and other gases, okay? So what do you think that structure of a biconcave disc helps them be able to do?
So they can move through the vessels?
That is one, so they can move through vessels, all right? You see it here, right up the top right here, up here. These is a transmission electron micrograph of a red blood cell that are flowing through vessels, okay? Let me ask another question. How does oxygen and carbon dioxide get on, what we call get loaded onto hemoglobin? Where does it happen? Where does oxygen get pushed into the blood? It's in the capillaries. I'm thinking more bigger, gross anatomy terms. Joe? In the lungs. And then how does it happen? Let's do it this way. So if we have, here is your lungs. Okay, it's not the best, right? But inside the lungs, you're going to have air pockets, and then you're going to have these little things called alveoli. And inside there, you're going to have this open space, and this is where inspired air comes in. So oxygen's in here. Then around this alveoli, you're going to have a blood vessel. Okay? A blood vessel. And blood runs through that. Red blood cells run through that blood vessel. Here's your concave discs, okay, that are running through there. How does oxygen get from here to there? What do you think? Is oxygen a small molecule? It's just two oxygens, right? Yes, it's relatively small. It's not as big as a protein. Is it charged? No, it's pretty inert. It's not polar. Small, not polar. All right. What do you think it does? It diffuses through. Okay? So it's going to diffuse from the inside the alveoli to onto the hemoglobin in blood. Okay? So now that's the process of how we load oxygen onto red blood cells. How is the structure of a biconcave disc aid in that process? What do you think? What does diffusion depend upon?
The concentration.
All right, so it's a concentration gradient. So you could say the concentration of oxygen is higher here than it is over here, right? So it's lower O2. So it's going to move this way, right? Okay. Now what else? We want how fast does blood run through capillaries? Pretty fast, right? But it slows down, and the capillaries are really small, and it slows down blood flow so that only the width of one red blood cell can squeeze through. That's what you see here as well, right here, right? So now think about the structure. Look at the structure of this red blood cell over here, okay? Look at the structure of this red blood cell. What do you think it does? What's the other part that helps to make diffusion an efficient process? What about surface area? Does that help? Yeah. All right. If you have a small cell with just a little bit of surface area, I'm sure in Bio 150, and I don't know about it. I know I've taught my general bio course. I talked about, when I taught mine, we went through why cells are small, right? They have a high surface area to volume ratio. What do you think this has? It has a high surface area to volume ratio, right? The higher the surface area to volume ratio, the faster diffusion can occur. Do we want diffusion of oxygen to occur at its fastest rate right here as the blood is flowing through? Yes, we do. Okay? So that helps that process. Okay? So high surface area to volume ratio. Let's do this. Get that out of the way. There we go. Okay? And with that structure relationship, so we have this high surface area to volume ratio. Let's write that down. Okay, we also have basically an ejection of organelles, no nuclei, no mitochondria, right? A lack of a cytoskeleton, or a lack of a rigid cytoskeleton. All right? Those things allow diffusion to occur fast, the high surface area to volume ratio, and then with that, we've replaced all the organelles with hemoglobin. Because what is the purpose of a red blood cell? What's its function, Alex? Transport oxygen. And what molecule inside the red blood cells does that? Hemoglobin. So the more molecules of hemoglobin I can pack into a red blood cell, the more molecules of oxygen it can carry, right? So essentially, erythrocytes are bags, cellular bags of hemoglobin, right? Again, they're going to make their function to be the most efficient process that they can. Okay, here's another diagram showing you a sectional view, and it says right there, there's about 280 million hemoglobin molecules per red blood cell. You saw how many red blood cells you were present in your blood smear on this week in lab, right? How many of you have not had lab yet? Right, there's no Friday afternoon lab. Is there? I don't know. If so, that sucks. Okay? And then what you end up seeing is that the size and the structure of these, right, they're about the diameter, all right, of the smallest capillary, so that a red blood cell can be able to fit through, right? And the size and the structure also allow them to stack. And that's what you call a rouleau right here. Right, see these cells right here? They're stacked on top of one another, so it looks like this. That's a rouleau right there. Okay? Relationship of structure and function. Are we clear with that? So now, if you end up changing the structure of the red blood cell, what happens to its function? Joe? It can't carry as much oxygen, or it doesn't deliver oxygen to the right way. Okay? And what you see in the bottom right left-hand corner here is an example of what? You looked at it in lab.
Sickle cell.
Sickle cell trait, sickle cell red blood cells, which lead to sickle cell anemia. You change the structure of the red blood cell, you end up changing, really what's depending upon the structure, changing the structure of the hemoglobin inside the red blood cell that causes this change in red blood cell shape. All right? Now this red blood cell, that's kind of sickle cell, right? Sickle in shape. It doesn't flow through capillaries easily. They can clump together, right? And then on top of that, the hemoglobin that's inside there doesn't bind oxygen as well. Okay, so if diffusion is disrupted, flexibility is disrupted, now you cannot deliver oxygen as efficiently as someone who doesn't, or as a normal shaped red blood cell. Structure and function relationship. Questions? Clear as mud. Good. By your silence, we move on. So, the important molecule in these red blood cells is hemoglobin. Okay? Hemoglobin gives red blood cells their color. It's a red pigment molecule. Okay? Red pigmented protein. It's consisted of four peptide chains that are fused together. You have 2 alpha globin units, 2 beta globin units, so there's 4 globin molecules that are held together, and there's also a heme group, or heme groups. Each globin chain has a heme group with it, and that's where the iron comes in. Okay? You talked about iron. The role of hemoglobin is that its most significant role is to transport what? Oxygen. Okay? People are starting to get actively involved. We're going to get all of these people involved. Okay? It will transport oxygen. Okay? But it will also transport carbon dioxide. Okay? So we say it's oxygenated when oxygen is bound to hemoglobin. That's also, we call that, I'll do that in red, call that oxyhemoglobin. That's the term for it. Okay? Deoxygenated when some oxygen is lost, when it's not maximally binding oxygen. If carbon dioxide is bound to it, which it can happen, okay? It's called carb-amino hemoglobin. Okay? Carb-amino hemoglobin. Now, is that counterintuitive? Think about, does that function kind of oppose each other? if oxygen is going to bind hemoglobin and be transported, why does carbon dioxide also bind to it? It is, here's what it is. It is not a competitive, they're not competitors for each other. Okay? Oxygen binds to the heme group of hemoglobin. Carbon dioxide can be bound and is bound to a different area, not the heme group. Okay? So they don't compete for binding sites. All right? And really, what ends up happening is that when carbon dioxide is bound to hemoglobin, it actually increases the affinity, right? The ability of hemoglobin to bind oxygen. Right? So now what's end up happening is you have this carbon dioxide shield, a red blood cell is bound to oxygen, it goes to the tissue, and it unloads oxygen in the tissue. Right? And it picks up carbon dioxide. Now it's coming back to the lungs, and it has this really high affinity for oxygen, but there's not enough oxygen until it gets to the lungs, and it picks it up really quickly. That increases the efficiency of diffusion and binding as it goes through the lungs too. Okay? So we're putting all of this together in order to maximize the diffusion process that occurs in the lungs. And we'll go over this again when we talk about the lungs, the respiratory system, as we go through. Okay? Here's a question. What type of protein structure is best exemplified by the hemoglobin molecule? And crickets. Let's do this. Okay, let's do this. This is great. Let's do, so hemoglobin displays what level of protein structure? A, primary. So you could get ready. B, secondary. C, tertiary. And D, quaternary. E, I don't know.